In Leipzig, in the spring of 2010, a sequence appears on a screen. It is mitochondrial DNA, extracted from a bone fragment no larger than a grapeseed. The technicians expect one of two results. Either this is modern human, or it is Neanderthal. Those are the only options. Those have always been the only options. The sequence matches neither. 385 base pairs separate this DNA from ours. For reference, the distance between us and Neanderthals is 202. This bone belongs to something else entirely, something no one had predicted, something that had never appeared in any fossil catalog, any museum drawer, any evolutionary model published in the previous 100 years. And the bone itself, the physical object that contained this revelation, is so small that you could lose it between couch cushions. That is where this story begins. Not with a grand excavation or a dramatic discovery in the field, but with a data point that broke a model. The cave where the bone was found sits at the edge of the Altai Mountains in southern Siberia, overlooking the Anui River and a valley thick with birch and pine. Denisova Cave. It is not large. The entrance is wide enough to let in natural light, and the interior stays dry and cool, with high limestone ceilings and a central chimney that draws air upward. It is the kind of place that invites shelter, and it has been inviting it for a very long time. Russian archaeologists from the Institute of Archaeology and Ethnography had been working inside it since the 1970s. They knew it well. The sediment layers stretched back hundreds of thousands of years, and the cave had yielded stone tools, animal remains, bone needles, decorative beads, and occasional fragments of bone that were clearly human or close to it. David Reich, a geneticist at Harvard who later traveled to the site, described it as a magical cave. The trip from Boston took 48 hours and ended with a 10-hour van ride to reach it. Remote does not begin to cover it. For decades, the interpretive framework was straightforward. Neanderthals had roamed Western Eurasia. Modern humans had emerged from Africa and eventually replaced them. If archaic human bones turned up in Siberia, they were probably Neanderthal or early modern human. There was no third category. The human family tree, as understood in the early 2000s, had two main branches outside of Africa, us and the Neanderthals. The question was how those two branches had interacted, not whether a third branch existed. This assumption was reinforced by what we could see. Fossils, skeletal morphology, teeth and skulls and femurs that could be measured, compared, slotted into categories refined over more than a century of physical anthropology. If a species existed, we expected to find its bones. And if we found no bones, we assumed no species. The fingerbone fragment, catalogued as Denisova 3, was recovered in 2008 from Layer 11 of the East Gallery by Anatoly Derevianko and Mikhail Shumkov of the Russian Academy of Sciences. It was tiny. It looked like it could belong to a child. It was sent to Leipzig, to the lab of Svante Pääbo at the Max Planck Institute for Evolutionary Anthropology, the same lab that had just finished drafting the Neanderthal genome from bones found in Croatia. Pääbo's team was screening material from multiple sites. They almost did not notice this one. But the cave's internal temperature, which holds at 0 degrees Celsius year-round, had done something remarkable. It had preserved DNA in a state of quality that almost never occurs in remains this old. When Johannes Kraus, a member of Pääbo's team, extracted and sequenced the mitochondrial DNA, the data was clean, abundant, and wrong. Wrong in the sense that it matched nothing in the existing database. The mitochondrial lineage diverged from ours roughly 1 million years ago, far deeper than the Neanderthal split. Two things happened very fast. First, the team sequenced the nuclear genome, which told a different but equally startling story. The nuclear DNA placed this individual as a sister group to Neanderthals, not a million-year-old relic, but a population that had separated from the Neanderthal lineage around 400,000 years ago and then gone its own way. Second, they found something no one was ready for. When they compared this genome to living human populations around the world, one group stood out. Melanesians, people living in Papua New Guinea and surrounding islands, carried 4 to 6% Denisovan DNA in their genomes. This extinct population, known only from a finger bone in Siberia, had interbred with the ancestors of people living thousands of kilometers to the south. A species that left almost no physical trace had left a genetic signature across an ocean. The question that should have been asked decades earlier suddenly became unavoidable. Why had no one seen this coming? And the answer is uncomfortably simple. The tools we had been using to classify ancient humans were designed to read bones, not genomes. If a species looked similar enough to something already known, its bones could sit in a museum for generations without raising suspicion. Denisova 3, the finger bone, was originally catalogued as probably modern human. It was unremarkable to the eye. A physical anthropologist examining it would have had no reason to flag it as extraordinary. Only the DNA made it visible. By 2012, Matthias Meyer in Peibo's lab had developed a technique for building single-stranded DNA libraries that captured far more of the degraded fragments than any previous method. Using fewer than 10 milligrams of the original bone, the team sequenced the Denisovan genome to 30-fold coverage, meaning every position in the genome was read, on average, 30 times over. Meyer called it a genome of very high quality, one with fewer errors than most genomes sequenced from living people. That is worth pausing on. The most complete genetic portrait we had of an extinct human lineage came from a chip of bone you could balance on a fingertip. The work earned Pabo the 2022 Nobel Prize in Physiology or Medicine, awarded specifically for his discoveries concerning the genomes of extinct hominins. It was the first time a new human group had been identified entirely through DNA, without a single diagnostic fossil to guide the way. This is the mechanism that kept Denisovans hidden. Not a failure of effort, but a limitation of method. For over a century, paleoanthropology had relied on morphology, the shape and structure of bones. Morphology is powerful, but it has a blind spot. It assumes that genetic distance correlates with visible physical difference. Two populations can look similar and be genetically remote, or look distinct and be genetically close. The Denisovan finger bone looked human enough to ignore. The Denisovan genome was different enough to rewrite the tree, and the cave itself compounded the problem. Denisova Cave was not untouched when the geneticists arrived. It had been excavated for 40 years. The stratigraphy, the layered record of sediment, had been studied extensively, but dating it was notoriously difficult. Radiocarbon dating works well up to about 50,000 years, but much of the cave's occupation stretched far beyond that range. Without reliable dates, it was hard to know when different groups had been in the cave, and harder still to know whether they had overlapped. The physical record was rich. The chronological record was a mess. It took until 2019 for an international team, including geochronologists from the University of Wollongong in Australia and radiocarbon specialists from Oxford, to establish a reliable timeline. They used optical dating on the sediments and Bayesian modeling to combine multiple dating methods. The picture that emerged was staggering. Denisovans had occupied the cave from at least 250,000 years ago until roughly 60,000 years ago. Neanderthals appeared around 200,000 years ago and disappeared from the site by 40,000 years ago. Modern human DNA first shows up in sediments deposited between 60,000 and 45,000 years ago. 3 distinct human species, 1 cave. And if you have been following this channel, you know the most interesting part of any discovery is not the headline, but the method that made it visible. That is exactly what happened next. Elena Zavala, then a doctoral student at the Max Planck Institute, led the largest sediment DNA analysis ever conducted at a single archaeological site. Her team processed more than 700 soil samples from across the cave. They detected ancient human DNA in 24% of them. Not from bones, from dirt, from the microscopic traces that bodies shed into the ground over millennia. That study, published in Nature in 2021, gave researchers a resolution they had never had before: a genetic occupancy map of the cave stretching back 300,000 years. But the discovery that captured the most attention came not from sediment, but from a single bone chip found among over 2,300 unidentifiable fragments. Samantha Brown, a graduate student at Oxford, had been scanning these fragments using a technique called ZooMS, which identifies species through collagen protein fingerprinting. It is fast, cheap, and does not destroy the sample. One fragment came back as human. Specifically, its mitochondrial DNA was Neanderthal. That fragment was sent to Leipzig. And what came back from the nuclear genome was something that had never been observed before in any fossil, from any site, anywhere in the world. The bone belonged to a girl, at least 13 years old, whose mother was Neanderthal and whose father was Denisovan. A first-generation hybrid. The team nicknamed her Denny, specimen designation Denisova 11. Now, finding a hybrid is significant. Finding a first-generation hybrid in a sample of fewer than 24 sequenced ancient individuals is statistically extraordinary. It implies that interbreeding between these groups was not a freak event. It was routine, at least when they encountered each other in places like this cave. Viviane Sloan, who led the genomic analysis, put it precisely. She had known from previous studies that Neanderthals and Denisovans must have occasionally had children together. She never expected to find one directly. But Denny's genome contained another layer. Her Denisovan father carried traces of Neanderthal ancestry from hundreds of generations earlier, from a different episode of interbreeding far back in his lineage. And her Neanderthal mother was genetically closer to Neanderthals who lived much later in Western Europe than to the earlier Neanderthals found in the same cave. This single genome encoded evidence of population turnover, long-distance migration, and repeated interspecies contact, all from a bone chip that had been sitting in a box with thousands of other unidentified fragments. The implications kept expanding, and they did not stay confined to a single cave in Siberia. In 1980, a Buddhist monk exploring Baishia Karst Cave on the Tibetan plateau stumbled on a partial jawbone embedded in limestone. The cave sits at 3,280 meters above sea level, in a region where thin air makes sustained human habitation punishing. The jawbone ended up at Lanzhou University and sat unstudied for 3 decades. In 2019, a research team led by Fahu Chen, Dongju Zhang, and Jean-Jacques Hublin analyzed the specimen using ancient protein analysis. The collagen proteins matched the Denisovans from the Siberian cave. Uranium decay dating placed the mandible at more than 160,000 years old. This was the first confirmed Denisovan fossil found outside Denisova Cave, and it placed these people at an altitude that would incapacitate most modern humans. Later work recovered Denisovan mitochondrial DNA from the cave's sediment layers, confirming occupation from roughly 100,000 to 60,000 years ago. They had not passed through. They had lived there. That Tibetan plateau connection set up one of the most striking findings in modern human genetics. In 2014, Rasmus Nielsen at the University of California, Berkeley, and his team published a finding that connected the Denisovans not to archaeology, but to modern physiology. They sequenced a gene called EPAS1 in 40 Tibetans and 40 Han Chinese. EPAS1 regulates the body's response to low oxygen. In Tibetans, a specific variant of this gene prevents the overproduction of red blood cells at high altitude, a response that protects against blood thickening, stroke, and pregnancy complications. 87% of Tibetans carry this variant. 9% of Han Chinese do. When Nielsen's team searched global genome databases, they found this variant in only one other place: the Denisovan genome. The match was almost exact. A 32.7 kilobase segment of the EPES1 gene in Tibetans was statistically indistinguishable from the Denisovan version. The team calculated that it could not have been inherited from a shared ancestor. It had to have entered the modern human gene pool through interbreeding roughly 48,000 years ago, when the ancestors of Tibetans and Han Chinese were still a single population. Most of those people eventually lost the variant because it offered no advantage at low elevations. But the ones who moved on to the Tibetan plateau, where the air holds 40% less oxygen, kept it. Natural selection did the rest. Nielsen called it the strongest documented instance of natural selection in any human population. A gene inherited from a species we did not even know existed until 2010 now allows millions of people to live at 4,000 meters above sea level. And that gene is not the only Denisovan legacy running through living populations. Denisovan DNA contributes to immune function variants found across Asia. The Ata people of the Philippines carry the highest proportion of Denisovan ancestry of any population measured, roughly 5% of their genome. Aboriginal Australians carry substantial Denisovan sequences. A version of the MUC19 gene, involved in mucus production and eye protection, was traced in 2025 to a Denisovan origin and found at high frequency across indigenous American populations. The genetic footprint of this species stretches from Siberia to South America, from sea level to the roof of the world. Meanwhile, in a separate line of investigation, the Denisovan genome revealed something deeper still. About 4% of its DNA does not match any known human species. Statistical modeling suggests it came from a vastly more ancient lineage, one that diverged from the modern human line more than 1 million years ago. Researchers call this the super-archaic contribution. The leading candidate is Homo erectus, which entered Eurasia roughly 1.8 million years ago and survived in some regions until surprisingly recently. But no Homo erectus genome has ever been sequenced, so the identification remains circumstantial. What is clear is that the Denisovans themselves were a product of deep, layered interbreeding with populations that stretch back toward the very origins of the genus Homo. The debate now is not whether Denisovans were real. That question is settled. The debate is what they were. Were they a separate species? A subspecies of Homo sapiens? Something taxonomically novel? For 15 years, no formal species name was assigned because the fossil record was too thin. A finger bone, a few teeth, a toe bone, a jawbone, and scattered fragments are not enough to satisfy traditional taxonomic standards. That changed in 2025. Qiaomei Fu and her team at the Chinese Academy of Sciences extracted mitochondrial DNA and analyzed endogenous proteins from the Harbin cranium, a massive, well-preserved skull found in northeastern China in 1933. The skull had sat at the bottom of a well for decades before reaching scientists. When the molecular data came back, it matched the Denisovans from the cave. For the first time, researchers could connect a face to the genome. The skull is low and long, with heavy brow ridges, wide eye sockets, and a large brain case within the modern human range. A September 2025 study proposed that remains from 6 additional Chinese sites, including the 1-million-year-old Yunxian fossils, belong to the same species, potentially extending the Denisovan lineage far deeper into the past than anyone had imagined. But not everyone agrees with that interpretation. Some paleoanthropologists caution that morphological similarities across such vast time spans could reflect convergent evolution or shared ancestral traits rather than species continuity. The Yunxian connection in particular is contested, since linking fossils separated by 800,000 years under a single species umbrella requires evidence that not all researchers consider sufficient. Convergent evolution and Yunxian are at the heart of the debate. The Harbin cranium itself was previously classified as a potential new species, Homo longi, and the reclassification to Denisovan is still being debated. Taxonomy aside, though, the molecular evidence from Harbin is strong. The genome belongs to the same population as the cave, and even as the picture sharpens, it remains fundamentally incomplete. We still do not know what Denisovans sounded like, how they moved through landscapes, what their social structures looked like, or why they disappeared. We do not know the identity of the super-archaic population that contributed 4% of their genome. We have never recovered Denisovan DNA from Africa or Europe, so we do not know the full extent of their range. The sediment DNA techniques that revealed the cave's occupancy history are still being refined, and every new site tested produces surprises. Full extent and sediment DNA continue to challenge researchers. The Denisovans were identified in 2010. In the 16 years since, they have gone from a data anomaly on a screen in Leipzig to a population whose genetic legacy is measurable in the blood of living people, in the lungs of Tibetan herders, in the immune cells of Pacific Islanders, in the eyes of indigenous Americans. They were never absent from the human story. They were absent from the methods we use to read it. Genetic legacy and the work are far from complete. The work is not finished. The oldest Denisovan genome, from a molar dated to roughly 200,000 years ago, was reported just recently, and it carried 5% of its DNA from a Neanderthal population that had never been detected before. Every new sample rewrites the model. Every new technique reveals another layer. We are still digging, literally. So here is what I keep coming back to. If the Denisovans were invisible for a century because our methods could not see them, what else is sitting in museum collections right now, cataloged as something familiar, waiting for someone to run the right test?